Hi, my name is Victor, and this is my concept map about plasma membrane. So I have four different um, characteristics of the plasma membrane that I would be going that I separated my topics over. First was the phospholipid bilayer, which is the the actual phospholipids. There's two layers on top of each other, kind of like a sandwich model, which was the first, one of the first models devised trying to explain what the plasma membrane was. Then it was the fluid mosaic model was later discovered, which proposed that also proteins and sterols and lipids and other organic proteins were attached to the plasma membrane, not just the phospholipids itself. This was proposed by Singer and Nicholson. Um, so the phospholipid bilayer is it's like I said before, two sets of phospholipids with with a head pointing towards the outside and the tails pointing in, so they're kind of opposites. This is because the phospholipids are amphipathic, which means that they have a group that is hydrophobic and hydrophilic. Hydrophobic would be the tails, that's why they're pointing away from the water, and the heads would be hydrophilic. That's why it would be pointing into the aqueous environment. You also have detergents. They're also amphipathic molecules, but they only have one tail when compared to phospholipid that has two tails. The tails of the phospholipids can be saturated, which means that there is no more carbons in that tail that can be bonded to another hydrogen. So it's saturated with hydrogen. And unsaturated, meaning that it has a double bond or triple bond. This bond makes the molecule not be straight, it kind of like bends. So if it has a double bond, it means that it isn't saturated with hydrogens. The other part, the head, which is hydrophilic, can bond to a lipid and create a glycolipid. Now going to the concentration gradient, which is kind of what the phospholipid lets. That's what the phospholipid kind of dissipates water towards or against it. So you have an isotonic solution, which would be a solution that is in equilibrium with the concentration of inside the cell to outside of the cell. Hypertonic, which would be the concentration of the cell is lower than the concentration outside of the cell, and hypotonic, which the concentration in the cell would be higher than the one outside of the cell. Now going to selective permeability, which is what the main function of the plasma membrane. It lets some stuff in and some stuff it does not let in. So you have two types of transports: passive transport, which does not require any energy, and this can be osmosis, which is the movement of water, and passive channels, which are channels that are a certain size or shape to only let certain particles in. You have active transport, which does require energy, which are like membrane channels, or gate ion channels, or ion channels, which require charged particles like potassium or sodium to be opened or closed, and these have to be charged, which are called electrical gradient. I'm going into membrane proteins. These proteins serve different functions. They can be transporter proteins, which can bring stuff into into the cell, kind of like membrane channels. That's one of the transport proteins, a receptor protein, which can receive a molecule and then send a signal to the rest of the cell. Anchor proteins, which anchor down filament or other proteins that are needed alongside of the membrane, kind of like an integral protein could be holding a peripheral protein. You have an enzyme protein which catalyzes reactions alongside of the plasma membrane. So you also have protein attached associated protein, just kind of with like an anchor protein. It doesn't have to be anchor. That's one example. It can also be a protein that's embedded into the membrane, and then protein, another protein is covalently bonded to it. That would be called a peripheral protein. Monolayer proteins are proteins that are only embedded into one side of the phospholipid bilayer. So it's only, you have two chunks, only in one chunk they would be bonded into. If any part of the protein is embedded into the plasma membrane, those proteins are called integral proteins. Lipid/sugar linked associated proteins are proteins that are bonded to sugars or lipids. For sugars, you have glycoproteins, which are proteins that have oligosaccharides bonded onto them, which they are around 12 to 20 monosaccharides long, and these are usually on the external matrix of the cell. They're not in the inside. And then also goes for protoglycan, which there are proteins that have a lot more monosaccharides, which are called polysaccharides, which is around 20 or more monosaccharides. And yeah, that would be it.